So now that we've looked at general information about nucleic acids, we're going to look at some specifics regarding the structure of DNA. But first we need to look at the history of how we discovered that structure of DNA. There were many people involved in discovering the structure of DNA. There was kind of a race to determine this really interesting information. Rosalind Franklin, who you're looking at, she was using some really new technology for her time called X-ray crystallography. What she found was an X shape. Here's her picture that she essentially developed using this new technology, X-ray crystallography. And how to read this as an X shape or a helix shape is actually very complicated, but she was able to determine based on her diffraction pattern here that DNA has a helical shape. It's like a twisted ladder of sorts. But in addition to knowing that DNA looks like a twisted ladder, there's a lot of other information that was needed. The man you're looking at now is Erwin Chargaff. He was doing some additional research based on what DNA was made of. And what he found was a very interesting pattern. So we looked at different nitrogen bases, and as you can see at the top here, you've got A for adenine, T for thymine, G for guanine, C for cytosine. And on the left, you have different organisms. So Streptococcus is a bacteria, yeast is a fungus, herring, that's a type of fish, and then human, which most of us are human. So what he found was a pattern in the occurrence of how much adenine you have for how much thymine you have and how much guanine and how much cytosine. And after doing the research, he found that the amounts of adenine and thymine were often very close. Now, 29.8 and 31.6 might not seem very close. However, compared to guanine and cytosine, which are closer to 20, and these are percentages, closer to 20%, those are more equal to each other, while adenine and thymine are more equal to each other. And that's true in every organism. So 31.3, 32.9 are closer to one another than 18.7, 17.1. And so this is something that he found in every single organism, even though from organism to organism or from species to species, the numbers can vary. The trend is the same in that you essentially have about the same amount of adenine as you do thymine and the same amount of guanine as you do cytosine. So this led to the development of Chargaff's rule, and that is that adenine is always going to bond with thymine and cytosine is always going to bond with guanine. Now, of course, this is in DNA. Remember, we looked at another nitrogen base called uracil, but that one's only found in RNA. Right now we're really looking at Chargaff's rule because we're talking about pairing. And if you remember the differences between DNA and RNA, RNA only has one strand, so it's not usually pairing with anything. But in DNA, adenine bonds with thymine, guanine bonds with cytosine. Something else that you really need to know is that adenine and guanine are slightly larger nitrogen bases. They are purines, while thymine and cytosine are pyrimidines. And the rule is that a pyrimidine will always bond with a purine. Okay, so you would never have two purines bonding together or two pyrimidines bonding together. Ultimately, you would have kind of a stranger shape in your DNA molecule, which can result in something called a mutation, which we'll talk about later on. But you do need to know that adenine and guanine are purines and thymine and cytosine are pyrimidines. And if I had to ask what was uracil, since uracil replaces thymine, it must be similar to thymine. So uracil is also a pyrimidine and it bonds with adenine. Now, all this information about DNA being a double helix and adenine bonding with thymine, guanine bonding with cytosine, all that information actually had to be put together to make a kind of cohesive idea about the structure of DNA. And the two folks you're looking at right now are James Watson, who's the young guy on the left, and Francis Crick, who's the older gentleman standing up. And these two are essentially responsible for putting all of that information together. You can see their little model that they created. They did get the Nobel Prize for determining the structure of DNA. And they did that by essentially getting information in less than ethical ways from Rosalind Franklin and Erwin Chargaff. We'll talk about that a little bit more in class because there's quite an interesting story behind how they obtained this information. But they did get the credit for developing the structure of DNA. And here you're looking at kind of a drawn out version of a double helix. I want you to notice that there are two strands. So if you're looking at this one strand and following it all the way across, this is one strand of DNA, and then here's the other strand of DNA. And notice that the way these two strands are bonded is with these base pairs. So adenine bonds with thymine, guanine bonds with cytosine. That's true as you continue to go all the way down the molecule. The other thing I want you to notice is that it's twisted. It's not just two straight strands that are attached to one another, and sometimes I might draw it that way, but in general it's also twisted. Okay, so it's the double meaning it has the two strands, and it's a helix, and helix means it's twisted. And in addition to understanding that it's a double helix, again, two strands that are twisted, what I want you to notice is what holds these two strands together. So what holds this strand with this AGT together to this strand with the TCA? And the bonds in between those nitrogen bases are hydrogen bonds. You might remember that from when we were talking about water. And now we're looking at it in the context of DNA. The two strands of DNA are held together by hydrogen bonds. Now, keep in mind that just because we're talking about the nitrogen bases being held together by hydrogen bonds, there's still two other parts to each nucleotide. Remember that here you've got a nitrogen base, but part of a nucleotide is also the sugar and the phosphate that are attached to that nitrogen base. And that's what this is referring to right here. The backbone, the thing that is actually holding together an entire strand of DNA, is that sugar phosphate backbone, and it is repeating sugars and phosphates. And we'll look at that in more detail once we look at DNA replication, which is how we would make more molecules of DNA. But right now, that's about all for structure of DNA.